Hello, in this video we will discuss about the toll-like receptor TLR. Toll-like receptor is the receptor which part of the innate immune system. For example, macrophage and dendritic cell are natural killer cell. These all type there are many type of toll-like receptor which that is part of the innate immune system, not adaptive immune system. But this toll-like receptor is responsible for the activation of the innate to adaptive immune system. So the toll-like receptors is the receptor which part of the innate immune system, for example macrophage, dendritic cell and natural killer cell. So on the other hand, the next is the toll-like receptor for in identification and gives the signaling pathway to activate the certain gene of macrophage. For example, macrophage and dendritic cell antigen presenting cell to give response. So after this, the toll-like receptor is the different type of toll-like receptor, but the basically is the 10 toll-like receptor are present, while the 10 is not understanding, not working under the research, but the toll-like receptor 11, which that is the uropathogenic wheat in mice, while the other are present in the human. So the toll-like receptor activation to the third line of defense, adaptive immune response. So let's begin to understand the first general concept about the toll-like receptor mechanism and their response. So in this way, basically the infected cell, when the antigen will invade into the human machinery, human body, so when inside the human body, it will be infect the different type of tissue. So when it will be if infect the different type of tissue, for example, this is the one tissue type which contain one cell is infected but the other is not. So this is the basic function which that is basically the infected cell will be occur due to the invasion of the pathogen. This is the pathogen, a virus. So after the infection, that is also produced the tumor necrotic factor alpha. That's tumor necrotic factor alpha and interleukin, which that is and tumor necrotic factor gamma, which that is used for the binding with the itself cell, the autocrinally to kill that cell, while on the other hand it will be moved into the blood circulatory system. It is the primary inflammatory cytokines. These primary inflammatory cytokines will be moved into the blood circulatory system and target to the different innate immune cell, for example neutrophil, and that is the proliferation, and monocyte also proliferation to form a macrophage. After the macrophage, and toll-like receptor will produce in the macrophage the toll-like receptor. So the toll-like receptor will be produced, and this will lead to go to the infection site. So this is the positive chemotactic movement, so the chemoattractant, and in this way the macrophage will move toward the infection site and detect through toll-like receptor of the for virus or antigen. After the detection, it will lead to cascade of phosphorylation as well as endocytosis. So the cascade of phosphorylation will produce the different type of for example, this is the antigen presenting cell. If so, it will lead to basically lead to activate the innate adaptive immune response. But in this way, this is the toll-like receptor we detect and endocytosed. After this endocytosis, so the toll-like receptor 3, 7 and 9 is used for the neutralizing of that pathogen. So in this way, you can understand after the detection externally through toll-like receptor will lead to cascade of phosphorylation events, which that will respond to produce the interleukin as well and tumor necrotic factor alpha. After this, here is the nucleus, and this is the toll-like receptor which that is present, and this is the extracellular matrix and intracellular matrix. So the extracellular matrix contain a outer dimeric form of the toll-like receptor and monomeric form of the toll-like receptor, which they are different type. This is the toll-like receptor 1 and toll-like receptor 2 combined together, and this is known as the heterodimer. While the heterodimer another is the toll-like receptor 2 and toll-like receptor 6, which that is formed. And on the other hand, the lipopolysaccharide and gram positive, which that is the toll-like receptor 4. After the toll-like receptor 4 will be bind, and in this way the toll-like receptor 5, which that will be detect and the flagella through here is you can see the toll-like receptor 2 and toll-like receptor 1 heterodimer formation, which that is used for the detection of the peptidoglycan, bacterial, gram-negative bacteria, and lipoprotein and zymosin fungi. This will also lead the toll-like receptor 2 and toll-like receptor 6. This that is also lead to detect these things. And the toll-like receptor 4, the homodimeric toll-like receptor is used for the detection of the lipopolysaccharide gram positive. And the toll-like receptor 5 will detect the flagella of the bacteria or other protozoa. And on the other hand, the toll-like receptor 11 used to detect the uropathogenic wheat in mice, uropathogenic bacteria and wheat in mice. So it is in the wheat. So here is you can see the virus, fungi and bacteria and double-stranded RNA and CpG DNA and single-stranded DNA containing virus will be enter when it will be endocytosed. After the endocytosis of this virus, so the inner side the toll-like receptor 3, toll-like receptor 7 and toll-like receptor 9 will work. For example, the toll-like receptor 3 will detect the DNA, single-stranded, double-stranded RNA, and the toll-like receptor 9 and toll-like receptor 7 will detect the single-stranded RNA and CpG DNA respectively. So after this detection from both mechanism, externally or internally, will lead to cascade of phosphorylation events, which that will lead to activate the certain transcriptional factors. So the certain transcriptional factors will be activated, and that will lead to uh, produce the transcriptional factor can be activated protein 1 interferon regulatory protein, which that will regulate the interferon production. Interferon is used for the neighboring cell activation and neighboring cell immune activation. For example, there are different types. Interferon means that it interfere. So the necrotic factor kappa B is also produced used for the apoptosis, and this necrotic factor kappa B will work paracrinally or autocrinally. So the activated protein and interferon regulatory factor and the necrotic factor kappa B, which that will lead to transcribe into the messenger RNA, and this all data will transcribe into the messenger RNA, and after this messenger RNA basically will move into the cytoplasm. After the movement into the cytoplasm through nuclear pore, so it means that interferon regulatory factors, which that will be translate, and this translation is basically it will be interferon regulatory factor will that produce and the activated protein 1, which that will be activate to differentiation and proliferation and apoptosis. It is used for the activated protein 1 is used for the differentiation, proliferation and apoptosis, while the interferon regulatory factor will be translate into the necrotic factor kappa B and interleukin 1b and the pro-inflammatory cytokines, the tumor necrotic factor alpha. This will lead to translate, and in this way the translation, the proteolytic cleavage for pro-interleukin 1b and pro-interleukin 1b and interleukin 18, that will lead to maturation and the pro-interleukin 18 and release as cytokines. This will lead to release of cytokines and the tumor necrotic factor alpha, enhance the immune response. This will lead to enhance the immune response, the caspase 1. This caspase 1 is basically the pro-interleukin 1b and pro-interleukin 18 will be caspase used for the cleavage formation. After this cleavage formation, the maturation and the release as cytokines. This the interleukin 1b and interleukin 18 is used for the as a cytokines, and this is used as a cytokines, and that will be bind with the different immune cells, and after this binding with the different immune cells and go into the extracellular matrix toward the into the circulatory system as well as other things. So in this way, the tumor necrotic factor alpha enhance the immune response. And let's begin to understand that interleukin 1b and interleukin 18, and there are other inflammatory cytokines which that will be produced, and it is used for the differentiation, proliferation and apoptosis. Why apoptosis will be occur? Because the apoptosis is important for the macrophage due to the neutralization of the pathogen. So here is you can see the monocyte, neutrophil, macrophage and T cell, that is the adaptive immune cell as well as innate immune cell collectively will bind with the interleukin 1b and interleukin 18. That will be bind to trigger and move toward the infection site. For example, this is the tumor necrotic factor alpha, which that will be bind itself with the macrophage is at autocrinally or paracrinally to other tissue. After to other tissue, that will cell infected cell will be die, and on the other hand the other cytokines which that will lead to tumor necrotic factor gamma, which that will be bind, and some other cell which that will be bind, and in this way the infected cell will be killed by apoptosis and the protection will be occur, and in this way the signal to other cell about protection and defense. So that is the major mechanism of the toll-like receptor. Thanks for watching. Please make sure to subscribe. Bye.